Good afternoon, everybody, and welcome to Webinar Wednesday. Today, we have a guest presenter, Peter Harris. And so during Dr. Harris's presentation, please be sure to type your comments and questions into the chat panel on the right side of your screen, or you can tweet us at PKD Foundation. Using the hashtag Webinar Wednesday. We'll do a Q&A at the end, just like usual, to answer as many questions as we have time for. I do want everyone to note that we, as usual, do not offer medical advice, and the information shared this afternoon is not intended to be a substitute for your a visit with your health professional team, your physician, your nephrologist, anything like that. So just know that this is general information for your general knowledge, but not intended to be a substitute. I am recording this webinar and will archive it on the website within the next few days. Okay, so like I said, today our guest speaker is Dr. Peter Harris, and he is a professor of biochemistry and molecular biology at the Mayo Clinic in Rochester, Minnesota. His research focuses on genetic diseases of the kidney, most specifically PKD. Dr. Harris has had a profound impact on PKD research, having identified the major gene for ADPKD and ARPKD among his many contributions. He is the director of the ADPKD Mutation Database at the Mayo Clinic and was a member of our Science Advisory Committee from 2004 to 2009. Dr. Harris is a huge friend of the Foundation's, and we are really excited to have him here today to talk to you about the genetics of ADPKD and ARPKD, how these genetics can be used to inform disease progression and even potentially drive therapy development. So with that, I'm going to pass this over to Dr. Harris and let him take it away. That looks great.
Is it okay? Can see okay?
Yep.
Okay, so I'm going to talk to you today about genetics of polycystic kidney disease. In particular, to talk about ADPKD and ARPKD. Okay. So if we consider ADPKD, so autosomal dominant polycystic kidney disease, or ARPKD, autosomal recessive polycystic kidney disease, these are in a group of genetic disorders that we consider as Mendelian or simple genetic disorders, where the gene mutation itself determines whether a patient develops disease. There's other genetic diseases where genetics also play a role, like diabetes, hypertension, and cancer. But in these cases, environmental and lifestyle factors play a much bigger role, whereas those roles are smaller for ADPKD and ARPKD. So I think it's worth thinking that we're all made up of lots of cells. From this analysis, 37.2 trillion cells, and each of these cells is really a self-contained organism in itself, which contains all the genetic material which is required to encode the proteins of the body and able to replicate itself. On this little picture of the cell here, I added a cilium because we know that this is important in the PKD disease. And it's worth noting that in the nucleus here, where the DNA is, we have about 2 meters of DNA in each of these 37 trillion cells. So if we think that this DNA is organized onto 46 chromosomes here, 22 of them which are the non-sex chromosomes, or the autosomes, and this is where the autosomal from autosomal polycystic kidney comes from, is to do with genes that are not sex chromosomes, but ones that we all have. And there's a pair of sex chromosomes, XX in females and XY in males. On these chromosomes, there's a total of about 21,500 genes. And the chromosomes are made up of DNA, and the DNA is the material that encodes the genes and then enables us to go on to encode the protein. Sometimes you hear the word genome, and the genome really is all of the DNA which is on all of the chromosomes, is what we're talking about when we say the genome. So if we think about this, then the total DNA, the genome, is really a library. The chromosome might be a book, and then each gene might be a page within that book. We have 2 copies of each of these pages or books, which come one inherited from the father, one from the mother. And then if we have a genetic disease, we have a mutation, which is basically a misspelling in this page. It's worth noting that there's lots of misspellings that occur all the time, but most of them don't occur in important parts in genes, so they don't lead to genetic diseases. So let's think about what a gene is. This rather extended explanation here from the Webster Dictionary. I think it might be better to break it down into little bits. So we can say, for instance, if we're talking about the physical description, it's a sequence of nucleotides, and I'll show you a nucleotide in a minute, in the DNA located on a chromosome. And then if we think about what the gene does, it works by specifying the structure of a particular protein, and we're all made up of proteins, so it's important that the proteins are made properly if our bodies are to work properly. And then the kind of explanation that Mendel, from the Mendelian inheritance, might understand is as a functional unit of inheritance controlling the transmission and expression of one or more traits. So in his case, whether P's were smooth or wrinkly, and in our case, whether or not you have a polycystic kidney disease. So this is the structure of DNA. Here we have the sugar regions on the outside here, and then we have the actual bases in the middle, the adenine, cytosine, guanine, and thymine. We usually consider them with just a single letter code. So most of the time we're just thinking about this sequence of DNA as encoding the gene and regulating the gene. So this is what a typical gene looks like in the human genome. It's about 27,000 base pairs long, and each of these pairs here, the T binds with A and the C binds with G here. So each of those base pairs, so we have about 27,000 of them that make up a typical gene. The gene is rather inconveniently broken up into pieces that code the protein and pieces stuck in between, which are called introns. And these places have to be taken out and the coding pieces put together before we make the messenger RNA and the protein. And we have regions at the start of the protein here and at the end of the protein which play a regulatory role in the expression of the gene product. So this is really the scheme of things where we have the gene encoded by the DNA. It is transcribed into what's called a messenger RNA. That RNA is then exported from the nucleus into the cytoplasm and translated into a protein. And it's the defective protein that we have in a genetic disease that is a problem. The protein is just not working properly. So that the code is a simple 3-letter code. We have 3 of these nucleotide pairs code for each different protein. And we have 20, or each different amino acid, sorry, and it's these amino acids that make up the protein. So we have 20 different amino acids that make up all of the proteins in our body, and we have 64 of these different triplet codons here, which encode these different amino acids. So there's some redundancy here. Often the third position in the code doesn't matter. Either any of them will encode the same amino acid. And then we have 3 special codes that tell the protein to stop. That's the end of the protein, end of the translation. So let's think a little bit about autosomal dominant polycystic kidney disease. You hear a lot of numbers bounded around by this, and I'll try and clarify or maybe muddy the waters about this. So I think an estimate of 1 in 1,000 having multiple cysts in the kidney is a reasonable estimate. I think probably an estimate of 1 in 2,000 individuals that is going on to reach renal failure, so requiring dialysis or transplantation due to ADPKD is a reasonable number. We know worldwide that 4 to 10% of patients with transplant or dialysis have ADPKD. The number is about 4.7% in the US. In 2014, nearly 32,000 ADPKD patients were on dialysis or had a transplant. In fact, about 2/3 of them had a transplant. So about 1 in 9,500 individuals in the country. At Mayo, about 14% of the transplant program is associated with ADPKD. As you know, this is generally an adult onset condition where 50% of patients experience renal failure by 60 years of age. And it's a progressive disease that develops over the lifetime of patients, with early cysts even developing during embryonic periods. So what do we mean by dominant? So this is a diagram of a family here, the parents and the 4 children. The father is affected, so the blue color here is showing the mutated gene. And we can see that this mutated gene is inherited here by the son and by the daughter. But the other 2 children don't inherit. They inherit the other copy of the chromosome from the father, so they don't inherit ADPKD. So this will be the typical situation where 50% of children within a family will develop ADPKD if there's an affected parent in the family. That males and females are affected equally, and the risk of having a SIB that's affected is 50% that SIB will be affected. So this is showing ADPKD pedigree here. So in this, this is just showing what a family looks like. So males are squares, females are circles, black ones are affected, light ones are unaffected. We can see the disease here is actually segregating through 4 generations in this family. If we look at this one SIB ship, so here's the parents here with an affected father, and we can see about half of the children are affected, both males and females, as we'd expect. So ADPKD is a very penetrant disease. We don't usually see the disease skipping generations, for instance. But just because you inherit the disease, it doesn't mean that the patient is going to develop end-stage renal disease. Some families look more like this, where there's evidence of the disease here. The mother has the disease here, but then her parents apparently are unaffected and she doesn't have any affected SIBs. And we know that new mutations or de novo mutations occur quite regularly in ADPKD, and at least 10% of families can be linked back to these new mutations. Sometimes the mutation doesn't occur at the in the sperm or in the egg, but can occur at a later time in the 4-cell stage in the developing embryo, as we see here. And if it occurs at this stage, the consequences are that the patient is actually a combination of cells that have the mutated gene and ones that don't have the mutated gene. And the consequences of this are that the disease is more variable, depending on if the kidney is made up of a lot of the cells with the mutation or not, will determine the severity of disease. This has only been rarely described in ADPKD, but in all patients that have a new mutation, this can be considered, and it's probably more common than we realize at this stage. So one of the questions over the years is, why do we develop a CIS if we already still have one functional copy of the polycystin gene, or the PKD gene? And a classical idea here is that another mutation occurs within the cells, within the tubules in the kidney, and only when we have no functional gene here do we start to develop a cyst. However, there's an alternative hypothesis, and one that we prefer, and that is that this dosage reduction, 50% of the functional PKD1 protein, is in itself is a susceptibility factor to develop CIS, but other factors might be important, just chance factors, other kidney damage will determine whether CIS actually develop. That doesn't mean that there isn't other genetic and non-genetic factors going on in these cysts as they grow larger, which is giving them a growth or survival advantage. So these are the actual two genes which are the common genes that cause ADPKD, PKD1 here at the top, and this is the gene itself, and then this is the messenger transcript that's transcribed from this gene. So you can see now all the bits that are separated up now are in one contiguous molecule, so that the protein can be translated from this transcript. And there's a PKD2 gene here. This is a smaller gene. So PKD1 is a very large gene, or at least it makes a large coding region. It has a coding region of about 13,000 base pairs. There's about 3,000 base pairs for PKD2. PKD1 is a kind of complicated gene because it lies in a duplicated part of the genome. So most patients have mutations in PKD1, with around 15% in PKD2. And in the normal studies we do, we don't find the mutation from genetic analysis in about 7%, and I'll talk a little bit about that in a minute. This is just showing the duplication of the PKD1 gene. This is a method called fluorescent in situ hybridization, or FISH. So we're taking a piece of the gene here and we're just sticking it onto the chromosomes. And we can see here at the end of the chromosome, this is the PKD1 gene, but we can see further down on the chromosome there's more copies. And in fact, in humans, we have 6 pseudogenes. So pseudogenes look like genes, but they don't really work as genes. And we can see that in chimps and orangutan, we also have this duplication, or in gorilla and chimps, if we go back to orangutan, we actually only have the PKD1 gene. So in evolution, this duplication occurred quite recently. This duplication makes genetic screening for mutations more complicated than for normal genes within our genome. So we can have mutations of different types, and this is showing a diagram here of the different types of mutation. This is actually from the HALT PKD study that some of you may have been involved with. And we can have mutations that are predicted to truncate the protein. They can be frame-shifting deletions of a single nucleotide, so that all the amino acids afterwards are wrong because the sequence of the code has been put out. We can have a so-called nonsense mutation, where one of these termination codons gets put into the wrong place in the middle of the protein. Or we can have splicing mutations which alter sequences here, which determine whether this intron gets cut out properly, and this can lead to the intron being left in, or actually exons that are in this region being skipped. So this is a usually also leads to a truncating mutation in the protein. Another group of mutations are ones which don't truncate the protein, non-truncating mutations. So missense are the most common there, where one amino acid is simply substituted for another amino acid within the protein. And another possibility is an in-frame change. Remember I said that the code consisted of 3 nucleotides. So if we get rid of 3 nucleotides, it means that one amino acid is missing, but actually the phase of the protein and the rest of the protein is not disrupted. So in those cases, these mutations may not be as severe as truncating mutations. And this gives just an idea of the mutation spread that we have in both the PKD1 and the PKD2 gene. As I mentioned, we don't find mutations in about 7% of families, and these non-truncating mutations, sometimes which are not completely penetrant mutations, as I'll show you, about 34% in PKD1 and about 11% in PKD2. So if we look at the PKD1 gene, each one of these lines and numbers here represents an individual family. And you can see that mutations can be found pretty much throughout the entire coding region of the gene. And the mutations can be of all the different types that I just told you about. And I think it's reasonable to say that any fully inactivating mutation to PKD1 or PKD2 will lead to polycystic kidney disease. Unlike some other diseases such as cystic fibrosis, there's no common mutation in ADPKD. So no single mutation accounts for more than 2% of the population of ADPKD patients. And that's reflected in these new mutations that seem to be occurring all the time. We have a database here funded by the PKD Foundation that we're updating at this stage. And in the updated version of the database, we have over 1,800 different likely pathogenic mutations, accounting for nearly 3,000 families. We try to score these mutations or these variants to see if we think that they're pathogenic mutations, and also to determine whether we think that they're fully inactivating mutations. So, as you might know, a diagnosis in ADPKD is usually done by renal imaging rather than genetic methods, where ultrasound, CT, and MR are pretty reliable for identifying whether an at-risk individual has ADPKD. Imaging can also be helpful to determine whether a patient has more rapidly progressive or slowly progressive disease based on the size of the kidneys, and I'll show you an example of that. But molecular diagnostics can be helpful in cases, for instance, in a transplant donor when the imaging is not informative, or if there's a negative family history and we're trying to determine whether it's really ADPKD in the family. And then unusually severe and mild cases also can be helped to be resolved by doing genetic studies. Up to this time, genetic studies have been rather expensive in ADPKD, partly because of this duplicated gene, and partly because there's been a monopoly of providing clinical services in the US. But now the market has opened up more, and as I'll show you, there's newer genetic methods, which mean that I think in the future genetic screening will be much more widely used in ADPKD. The genetic information can also give some idea about the likely progression of the disease within the patient, both with the gene involved and the particular mutation. And I'll show you some indications about that. So this is the new genetic method, so-called next generation sequencing, and one popular method is whole exome sequencing. I don't want to go into this in detail, but Suffice it to say, instead of just screening one gene at a time, we can screen all of the coding regions in the genome, or we can screen a group of genes which are candidate genes, which makes it easier to do this analysis. The duplication of PKD1 is still a problem, but I think that problem is being overcome. So I think it's likely that there's going to be more available molecular testing, and it's going to be cheaper, and that's already occurring to some extent. So knowing the gene that's involved is an important marker of how severe the disease is going to be on average in the family. If it's a PKD1 mutation, this is a bit of an older study here, but here the average age at end stage renal disease was 54 years compared to 74 years for PKD2, as you can see in the survival plots down here. We can also look at the We can also look at the size of the kidneys using MRI, for instance, or CT, and this is also a good biomarker of how severe the disease is. This is data from the CRISP studies that some of you may have been involved with. When we can see here, this is a single patient here with analysis done over 4 years, and we can see that the development of the disease sometimes can be quite rapid with large increases in the kidney volume, although other patients have much more of a benign course of the disease. And consistent with the size of the kidneys being associated with the severity of the disease, we can see that on the whole, PKD2 patients in blue have smaller kidneys than PKD1 patients, although there are a group of PKD1 patients also with smaller kidneys. In this French study, they were able to divide mutations into ones that truncated the protein and didn't truncate the protein, as I talked about earlier. And they were able to show, again, that PKD2 is the mildest in red here. But they were able then to separate the truncating and non-truncating mutations. So in this study, truncating mutations resulted in renal failure on average at 56 years, whereas non-truncating at 68 years. So you can see that the type of mutation can have some influence on the severity of the disease. This is just showing an illustration of these 3 different groups of patients. This one with a truncating PKD1 mutation, you see very large kidneys here in a patient of 45 years of age and close to requiring dialysis or transplantation. This is an in-frame mutation, a missense mutation. You can see that the kidney disease is actually very much milder, although there's quite significant liver disease here. As you may know, liver disease is something that presents to some extent in most patients, but severe polycystic liver disease requiring resection or transplantation is also something that occurs rarely in ADPKD. The liver disease doesn't seem to be closely associated with the gene or mutation type. And on the end here, we can see a PKD2 patient. We can see that their kidneys are somewhat enlarged. This is height-corrected kidney volume here. But you can see that it's made up a rather small number of cysts compared to the truncating PKD1, and that there's still regions of the kidney which are relatively unaffected. And that's reflected here in the conserved renal function we see in this patient at 53 years of age. Rarely, ADPKD can present as a much more severe disease, similar to the recessive form of polycystic kidney disease, even in families that have typical ADPKD. And one way that this can happen is a combination of a mutation that's spread throughout the gene here in PKD1, but then a second variant in the same gene, and the combination of these 2 variants is resulting in this earlier onset of the disease. This gives rise to our idea that the dosage of the functional PKD protein, Polycystin 1, is what's really critical in terms of the severity of the disease. If we have one inactivating mutation, we have this adult onset disease. If we have a very weak mutation by itself, we just get a very small number of cysts. If we get one of these hypomorphic mutations, which is not completely inactivating, we might have disease like PKD2, even though it's a PKD1 mutation. And then if we have a combination of an inactivating and an in-frame allele, we can get this very severe disease. We know that if we have two inactivating mutations, this is not compatible with life. And then there's other factors, genetic and non-genetic factors that are influencing the severity of the disease. Recently, or last year, we identified a third gene for ADPKD, making it a little bit more complicated. This is a disease that's actually rather mild. We can see just a few cysts in the kidney and a few cysts in the liver in a family with this disorder. And now we published a paper last year describing 9 families, and we found about 20 families now that have this GANAB as a third cause of ADPKD. These patients do well renally. They have preserved renal function, although they all develop cysts within the liver. They have variable kidney. So all have cysts within the kidney. And then they have variable severity of liver disease, including a few patients with very severe disease like this that require a resection or a transplantation. This is not a common cause of ADPKD, probably accounting for 0.3%, but maybe more common in patients with significant liver and mild kidney disease. There's other factors that influence the severity of the disease, and as we have larger populations, we're able to tease these out a little bit more. If we look at a PKD1 population and we look at gender, we can see that male patients have quite a lot more severe disease, with typical renal failure about 6 years earlier than female patients. But there's also some studies suggesting that high salt intake, a higher body mass index also are negative factors in terms of the severity of the disease. Smoking is probably also not such a good thing for any of us, although there's not really good studies showing its influence in ADPKD. And in terms of moderate intake of alcohol and caffeine, there's also really not good studies at the moment showing whether that's that it's detrimental, and there's not really good studies showing that it's harmful. In this French study, they used a combination of genetic factors and other clinical factors such as hypertension before age of 35 or urological events before 35, as well as the sex difference we saw here, to come up with a score of determining whether a patient is going to have fast or slowly progressive disease. And here you can see it was possible to divide patients into ones with much more rapid disease, mainly PKD1 truncating mutations with early clinical events, and then much milder disease, so these would be more likely PKD2 or in-frame PKD1 changes without early clinical events. So this information can be helpful for determining how severe the disease is likely to be in a patient. We're interested in finding other genetic factors that may be associated with the severity of disease beyond the disease gene, using a so-called genome-wide association study. These are factors that are relatively common within the genome and have a rather small effect. And some of you may be involved in this ADPKD modifier study, where we collected 1,600 patients in a collaboration with many sites around the world. And now we're collecting 2,000 more patients to try to find genetic factors that beyond the disease genes that may be associated with the severity of the disease. And this is an ongoing study at this stage. So in other diseases, other simple genetic diseases or Mendelian genetic diseases, such as cystic fibrosis, the mutation itself has become a target for therapies. For instance, read-through of nonsense mutations is one possibility, although these haven't worked very well clinically at this stage. The types of drugs that are showing some promise are ones that help the protein to move to the surface, which is its normal location. And we know that some PKD mutations also don't traffic to the cilia properly, and that's maybe the problem. So these types of drugs or similar drugs may be suitable in ADPKD. And then there's also drugs that help the protein to work properly. And there may be also patients where this type of drug is possible. So at this stage, this type of analysis is in its infancy in ADPKD, but in the future it may be that patients will want to know not the gene that's involved and the mutation involved, because it'll open up treatment options for you. Although, as I say, this is only really just starting. I want to finish up by talking a little bit about the recessive form of polycystic kidney disease, ARPKD. This is also quite a frequent genetic disease, about 1 in 20,000 individuals. Typically, it's the more severe infantile form of the disease and associated with neonatal death. It's also associated with childhood end-stage renal disease and large kidneys in infancy. And the disease in the liver here is not cystic, but a fibrosis, so-called a congenital hepatic fibrosis. And this can become important in older patients with ARPKD. So here we're talking about recessive inheritance. So this time we need 2 mutations before we develop the disease, one inherited from the father and one inherited from the mother. And we can see the affected child here that's inherited these 2 mutated copies of the gene. We can see in the daughter here or the son that they're carriers. They have 1 copy of the gene, just like the parents here. And in ARPKD, carriers are not really affected, although occasionally they may have a few liver cysts. And then in this individual, they inherited both of the normal copies of the gene, and so this individual is not a carrier, just a normal individual. So 1 in 4 individuals, offspring in a family, will develop the disease on average. Mutations for ARPKD are quite common. About 1 in 70 people in the population is a carrier for such a mutation. As I mentioned, the risk of developing ARPKD in the child is 25%. If a child with ARPKD has a child, the risk of having an affected child is quite high because of this high carrier risk within the population. Genetic diagnosis is probably used more commonly in this disease because it's a more severe disease, and pre-implantation genetic diagnosis, where you use in vitro fertilization and then select embryos that are unaffected, is something that's increasingly being used to determine to avoid having an affected child. But obviously this is up to the family whether they think this is a suitable thing for their particular family. Here's the gene that causes ARPKD, PKD1. This is again actually a very large gene with a large coding region. And again, a wide range of different mutations cause this disease. These are missense ones, so in-frame changes. And down the bottom here, I'm showing frame shifting changes. And in-frame changes are quite common in this particular disease. This mutation here, this T36M, is actually quite a common disease, and in patients of European origin, about 15% of the mutations are this particular missense variant. As in ADPKD, the type of mutation is associated with the severity of the disease, and if you have, or if there's two fully inactivating mutations, this is not compatible with life. As in ADPKD, you have two fully inactivating mutations. So patients, surviving patients, tend to have one or two of these in-frame changes. Though we consider ARPKD a disease of infancy, it can also be a disease which presents later and is usually only clinically significant as an adult. Here's two patients from a family. We're showing here dilation of the bile ducts in this patient and some cysts within the kidney, although you notice the kidneys are not hugely enlarged. And then the sister here, she has this congenital hepatic fibrosis here, and although there's a kidney stone here, there's not really a cyst within the kidney. But from mutation screening, it's possible to show that this is a ARPKD family. So ARPKD is not always an infantile disease, depending on the particular variants or the mutations that are causing the disease. So just to summarize what I've tried to say to you today, ADPKD is a genetically heterogeneous disease with two common genes, PKD1 and PKD2, although a third gene associated with mild disease. PKD1 is more severe than PKD2 on the whole, and non-truncating mutations are also milder disease, and allele combinations can give rise to early onset disease. But genetic diagnosis can be helpful for living-related donors and also members with negative family histories or mild or severe disease, and in some other cases. And other genetic and environmental factors, although not strongly influencing, can influence the presentation and the development of the disease. In ARPKD, the one gene is PKD1 here. There's lots of different mutations, again, that cause this, although at least one more common mutation. And again, there's some influence between the type of mutation and the severity of the disease. And pre-implantation genetic diagnosis may be useful in this disease. So that's all I have. I wonder if you have any questions.
Yes, we have lots of really great questions that have come in. So I'm going to dive right in. One question, and we actually accepted questions via email ahead of time. So there's kind of one question that seems to be asked a lot, and that is skipping a generation. So some people have let us know, like, they have PKD, but their sister did not, and their father has it, and other people are saying, you know, their doctor told them that PKD can skip a generation. Can you comment on that, Dr. Harris?
That's not normally true. The disease presentation can be different in different members of the family. So you could have a parent and a parent dies of a car accident at a young age, or something else, and is not known to have polycystic kidney disease. But if the parents live to an older age and then the child has the disease, and then the grandparent has the disease, or the grandparent has the disease, it's unlikely that the parent in between will not develop the disease. As I did show you, though, there's a lot of families where there's not a straightforward, or there's a new mutation that occurs within the family. So in that case, the parent may not be affected, or they wouldn't be affected. Although the child and then their children might be affected. So we don't see normally skipping of generations. Sometimes, you know, we see complicated situations where there's more than one disease in the family, or somebody had kidney disease. A grandparent had kidney disease, but it turns out when we look at it in more detail that it wasn't actually polycystic kidney disease. So there's not normal skipping of generations, but new mutations and variability in how the disease presents and progresses can sometimes make it a little bit more difficult to trace the disease within a family.
Okay, so to follow up, because we also had quite a few questions on spontaneous mutations and people who have no family history. So just to kind of follow up, is there a when a patient has a spontaneous mutation or a new PKD mutation that results in their having PKD, does their genetics look the same as all of these other PKD patient genetics that you've been talking about, or is there something different about the spontaneous mutation that's unique to that situation versus one that is kind of inherited from a family trait? Does that make sense?
Right. Yes. So usually, if the new mutation has occurred in the sperm and the egg, and the mutation is in every cell of the patient, including their germ cells, so their sperm or egg, then the disease will be transmitted to the next generation, just as if the disease has been seen for several generations in the family. If it's a mosaic, as I showed you, where the mutation occurred at a slightly later stage, then the mutation may get into the germline and be transmitted to the next generation, and may not. So there are possibilities where the patient can have disease in this generation, but there's not disease in earlier generations, and then their children also don't have the disease just because the mutation didn't get into the germ cells. So normally, if the mutation is in the egg and the sperm, the segregation of the disease in the children and the 50% risk is accurate, but because of mosaicism, it doesn't always work exactly like that.
Okay. So someone is asking about the relationship between PKD and PLD, or polycystic liver disease. And we've also just had some recent results out of Yale that one of our funded fellows, Whitney Bessie, worked on about PKD and PLD genes. Can you comment on the relationship between those two, and can they occur independently?
Yeah, I guess we're changing our mind about this a little bit. We used to consider ADPKD and ADPLD, so where you got cysts in the liver but rarely cysts in the kidney, as completely separate diseases. But the new gene that we identified, GANAB, we can see that we actually have presentations that sometimes look just like ADPKD and sometimes just look like ADPLD, where we just get cysts in the liver and very few cysts in the kidney. And we know that probably these two diseases are linked by influencing how the PKD1 protein, polycystin-1, actually is processed and gets to the cilium. So although we generally consider these to be separate diseases with different genes associated with them, our ideas are changing a little bit with some ideas, even with some PKD2 mutations, weaker mutations, not giving rise to much kidney disease, but giving rise to liver disease. I mean, I'd say that anybody has sort of unusual presentations of the disease in their family. We're interested in doing research genetic testing, and if you get in touch with the PKD Foundation and they forward it on to us, we can get back in touch with you about doing research testing to answer some specific questions.
Absolutely. And we did have someone ask earlier, and a couple people, we actually have quite a few questions about, do you advise people to be tested, to have their, sort of their mutation figured out, and, what are the pros and the cons? I think I can say a lot of physicians that I've spoken to, nephrologists, will sometimes warn against testing, especially in a younger person, a child, or even someone in their early adult years, because once you are diagnosed with PKD, you may have a harder time or may be disqualified from getting any kind of life insurance. And so that is a concern to consider. But for a scientific, a therapeutic perspective, what do you think about diagnosing people, adults, maybe well into adulthood versus children and young adults? What's your opinion there?
So we don't usually recommend testing in individuals earlier than 18 years of age unless they become symptomatic for the disease. And that's so that the choice of being diagnosed is left with the child once they become an adult. I think as more clinical trials are being done, and patients that are younger patients where the disease is still not very severe, then there's maybe some reasons why getting a firm diagnosis at a younger age may be helpful. I think patients are making family decisions about having a family, and things like the pre-implantation genetic diagnosis is a reason why some other individuals are interested in being diagnosed. As Alexis says, there is some negative factors still, even with the laws in place protecting discrimination about genetic diseases for things like life insurance. And so certainly sometimes people want to get established in their life before they get a diagnosis. I think these things may change as more therapies become available, and patients want to take therapies at an earlier stage. But as you know, in the US, no therapies have been approved yet, although there's one therapy in many other areas in the world, and I think it's very likely that therapies will be available here in the next few years. And so that's another factor that may come into consideration, because we'll probably want to treat younger patients, so getting a diagnosis then. And obviously you don't need to have genetics to get a diagnosis. In many cases the imaging can be enough to provide a diagnosis. If in the future there's more gene or mutation based therapies available, then it will be necessary to get genetic testing to determine the type of mutation you have, to see if a particular therapy might work for you. So I think it's a kind of changing situation, but now we're still advising people to be fairly cautious, I guess.
Okay, great. So I will say now we have less than 10 minutes left and quite a few questions, so we'll get to as many as we can, and if we don't get to them all, I apologize. So I think one thing people are really curious about is, the difference between PKD1 and PKD2. So one person is asking how many people have a PKD2 mutation or a PKD2 truncating mutation, which I'm not sure quite that's quite right.
So
assess the disease population, disease progression, and just what should they be looking to figure out if they have one versus two, and is there an advantage to finding that information early for treatment and progression and things like that? So kind of a reiteration, I think.
Yeah, so on the whole, PKD2 mutations, whether they truncate the protein or don't truncate the protein, are associated with milder disease. So average age at renal failure in the late 70s. So certainly PKD2, a diagnosis of PKD2 is a somewhat reassuring one, and one where therapies may be less indicated. For PKD1, it's a little bit more complicated. PKD1 mutations that are predicted to truncate the protein on the whole are associated with more severe disease, so average age of renal failure in the early 50s. For the non-truncating mutations, we really have quite a a mixture there. If we look at the whole population, the disease is milder than for the truncating mutations, but some of those mutations can be as severe as a completely truncating mutation, and some of them can be very mild, even milder than we see in PKD2. So there's a real range of outcomes associated with these non-truncating mutations. And one thing that we're trying to do is to get a better handle on each of the individual mutations and make a better estimate of how severe the disease is going to be associated with that. Obviously, there's other genetic factors that are also influencing the severity of the disease. So the gene type and the gene mutation is not everything, but it is something that provides some guidance of how severe the disease is going to be.
Okay, great. And several people are asking, because you mentioned that if people are interested in doing this for research purposes or having their genome sequenced, that they could reach out. So if you want to reach out, you can send an email to research, R-E-S-E-R-C-H, at pkdcure.org, and I will be able to connect those emails with Dr. Harris, and then he can review and decide if it's something that would be beneficial. We will also, in our follow-up email, send some information about the modifier study that's happening at a couple of different places that would also get at the same end game there. If someone wanted to seek genetic counseling, family planning, things like that, where, would they just go to a general genetic counselor in their area? Do you think those people would know enough about PKD to appropriately advise them? Or how would you, advise our patients to move forward in terms of, looking for some genetic counseling in that regard?
Yeah, I think that most genetic counselors should be well informed about polycystic kidney disease and should be able to provide good advice. I think the majority of ADPKD patients probably don't ever see a genetic person, and I think they can, get very good advice from their nephrologist. It depends a little bit on whether the nephrologist is a specialist in PKD, and I think there is some advantages in seeing a nephrologist that's a specialist in PKD. They'll probably be able to provide more accurate information and more up-to-date information about the disease.
Okay. Okay, back to maybe one or two more questions here. One person is saying, or is asking, and I think you might have talked about this a bit, so just to reiterate, when you have a mutation in the same family, does that always mean the exact same mutation is passed along, so that everyone in a family has the exact same type of mutation?
Yes, normally it's the same mutation which is transmitted through the family, so transmitted from the parent to the child in the dominant form of the disease. So even if it's a very large family that could be traced through many different generations, it will be the same mutation that causes the disease. Although, we still see some variability in the way that the disease presents and progresses within the same family. Just very occasionally, we've seen situations where, there are two different mutations within the same family. We studied one where there was a PKD2 mutation in part of the family and a PLD mutation in a different part of the family. So, occasionally we see those situations, but certainly the rule would be that the same mutation is causing the disease within the family.
Okay, and then any variability would be due to the expression or the phenotype of that gene and not a different mutation itself.
Right, usually, yeah, that's right. I mean, but it could be, other genetic and non-genetic factors that they're modifying the phenotype, and that's the one thing that we're trying to do with the modifier study. Although we're actually not recruiting any more patients for the modifier study, we're near in the analysis stage. But that study is trying to find other genetic factors that are influencing the disease. And the advantage of having this large, well phenotyped and genotyped population is that we can start getting other ideas about other factors that are influencing the disease, other lifestyle factors, and gender, as I showed you before, that's perhaps having a bigger influence than we previously realized.
Okay, and I know we're at 1 o'clock, or yeah, 1 o'clock Central, but there's one more question and then we'll wrap up here, so I apologize, we're gonna go a few minutes over, but something that's come up a couple of different times is where do the sort of extra-renal manifestations fit in on a genetic level? So if you if you know you have a family history of brain aneurysm or maybe of some other, extra-renal manifestation, can you comment, is that something that happens on a genetic level, or is that more on a clinical and a disease level that isn't informed by genetics?
So if we consider severe polycystic liver disease, the influence of the disease gene seems quite low. We know that patients with PKD2 are as likely to develop severe polycystic liver disease as PKD1, although, as you might know, females are much more likely to develop severe PLD than males, about 80% of patients. So there's probably some hormonal influence there, and there's probably other genetic factors, but they're not associated with the disease. With aneurysms, I think that the situation is less definite. We know that in a family where there has been an aneurysm, the likelihood of another individual within the family developing an aneurysm is higher than in typical ADPKD patients. So there probably are genetic factors that are influencing that. Again, though, I think it's quite likely that there are factors beyond the gene itself that are influencing whether a patient develops aneurysms. And there are some studies now trying to identify these other genetic factors.
Okay, thank you so much. And I think at that we are going to wrap this up. I know that there are still some questions that weren't answered, so I will do my best to work with Dr. Harris to get some general questions and answers up on the website. I do think that we got to most of the themes of the questions, so even if I didn't ask your question specifically, likely I touched on the sort of the content of your question in another way. So thank you everyone again for joining us for this webinar, and Dr. Harris, thank you for spending your afternoon with us to talk about the genetics of PKD. Again, if you'd like to reach out to learn more about potentially getting your genes tested, you can reach out to me at research@pkdcure.org. And with that, I will say good afternoon to everyone and have a wonderful day. Thanks a lot. Bye-bye.